We're going to start this lecture by reviewing the types of immunity. Remember, there are the natural or innate, that's the same thing, and then adaptive. And within the natural or innate immunity, remember it is nonspecific. There is no memory involved. It includes both external and internal defense systems and acute phase reactants. Within the adaptive immune system, it is specific. It can be a memory response. It involves antibodies, T cells, B cells, plasma cells, and cytokines. So we had the first lecture that was on the overview of the immune system, immunity. Then we talked about the nature of antigens. Then we had a lecture on just the innate immune system, and this lecture is going to focus on adaptive immunity and the lymphoid system, which is an integral part of the adaptive immune system. So the adaptive immunity, there can be both active and passive. So you can have naturally acquired active immunity or artificially acquired active immunity. So what active immunity means is that the patient or the person is introduced to an antigen or some type of immunogen, and they have to actively make their own antibodies. So it's an active process. Passive immunity can also be naturally or artificially acquired. And passive immunity, the best example of that is if you're given an immunization and the vaccine just includes the antibody. Or an example would be RhoGAM. So if you are a Rh negative blood type and you're pregnant with a positive blood type baby, they give you RhoGAM, and that gives you passive immunity, so your immune system doesn't take over and make an antibody. So that's the difference between active and passive. And don't worry, we're going to talk more about that later. So to review, let's review the acute phase reactants. Those are going to be really important. Remember, C-reactive protein is an early indicator of acute inflammation. We're going to talk way more about complement, but it's an opsonin, a chemotaxin, and it is involved in cell lysis. Alpha-1 antitrypsin can see increases in emphysema or liver disease, but these are very nonspecific. The point of acute phase reactants is that they are nonspecific. So for example, if you have a positive or elevated C-reactive protein or erythrocyte sedimentation rate, that's not going to tell anybody if you're having a heart attack or not. It's just going to say that you have general inflammation and that you have some type of immune process going on in your body. Haptoglobin is the protein that binds free hemoglobin released during hemolysis. Fibrinogen is a coagulation factor that helps form clots. And then ceruloplasmin binds copper. Okay, now we're going to get into the lymphoid system, and we're focusing on the adaptive immune system now. I wanted to show you this picture of hematopoiesis because we mentioned this briefly in an earlier lecture. And so hematopoiesis is just the process by which blood cells, red blood cells and white blood cells, develop from a hematopoietic stem cell. So this hematopoietic stem cell at the top, that's a stem cell, and a stem cell has the possibility or the potential to become any cell in the body. So it depends on the exposure to different chemical cytokines, so cytokines are chemicals released by cells. So the pathway or the cell that a stem cell develops into depends on the chemical messengers present. So don't worry about this later. We're going to learn all about this in hematology, and you're going to love it. It's so interesting. But notice how we have the stem cell here. Then we have the myeloid cells over here. So this is going to be all the granulocytes that we talked about, neutrophils, eosinophils, basophils. And then over here we have the common lymphoid progenitor. And that's going to be our T cells, B cells, dendritic cells, and plasma cells, natural killer cells, so the lymphocytes. So very specific. The lymphocyte is the key cell in the lymphoid system's immune response. There are about 20% of these circulating in our overall white blood cell population. They are about 7 to 10 microns in diameter. So the diameter, the nucleus of the lymphocyte is about the same size as a healthy, normal red blood cell. They usually have a rounded nucleus that may be indented. And don't worry so much yet about the chromatin, but this is where we're going. Medical laboratory professionals are very detailed oriented, and so we're going to learn to train our eye on the details of even the cytoplasm and the nucleus. Like, what does the chromatin of the nucleus look like? What does the cytoplasm look like? The colors, any vacuoles, any nucleoli present. So focus on details. Make sure that you are detailed oriented. And these lymphocytes differentiate into T cells and B cells in the primary lymphoid organs, and that is the bone marrow and the thymus. So we're not going to spend a whole lot of time on this, rehashing this. We talked about this in the intro, but just so you know that it is important. It could be a test question, probably is. What are the primary lymphoid organs? Bone marrow, thymus. And where do T cells go to develop the thymus? Where do B cells mature mostly? Some are in the bone marrow, some are in the lymph nodes. We talked about the secondary lymph node organs already, the salt or the malt or the galt. And we talked about the spleen, that it's a filtering mechanism for antigens in the bloodstream. The lymph nodes filter fluid from tissues. The mucosal surfaces are on our respiratory or gastrointestinal tract. And the circulation of the lymphatic fluid is all over the body, and it's controlled by those cytokines. So cytokines are cytocell kines chemical. So if a cytokine is a cell chemical, a lymphokine must be a chemical produced by a lymphocyte, and that's exactly what it is. Here's a picture of where the primary lymphoid organs are, so you can have a visual. And I love this picture of the lymphoid system. To me, these look like a highway, and I imagine these lymph nodes as highway patrol hotspots where the patrol officers are hanging out waiting for the bad guys to come along. So in our body, we have the lymphoid system, and these lymph nodes are where our lymphoid cells like to congregate and hang out, and they are watching this filtered fluid come through, and they are looking for any foreign antigens that don't belong there so they can mount an immune response. There are 3 types of lymphocytes. This should be a review from the first lecture or so. T lymphs, B lymphs, and natural killer cells. And you will hear me say B cells, T cells, natural killer cells, lymphocyte. I'm talking about the same thing. T lymphocytes are effector cells that serve a regulatory role, while B lymphocytes mature into plasma cells and produce antibodies. Both T and B cells recirculate continuously from the bloodstream to the secondary lymphoid organs and back. And this is done to increase contact with foreign antigens, because remember, they're like little police cars. They're surveying our whole body all the time to see if they can catch a foreign antigen. Natural killer cells are very large, they're more granular, and they play a role in both the innate and adaptive immune system. So this is kind of about the adaptive immune system, but just remember that the lymphoid system is involved in both innate and adaptive if you're talking about natural killer cells. The formation of lymphocytes in the bone marrow is antigen independent. Okay, and this is good because it would be a travesty or a tragedy if we only made lymphocytes when we already came into contact with a foreign antigen. The bone marrow is considered the largest tissue in the body, and it fills the long bones. And this was a trivia question. B cell maturation takes place in the bone marrow, and in chickens it's called the bursa of Fabricius. Now, I don't know why that's a fun trivia fact, but it was in your book, so I thought, hey, I'll add it. And lymphocytes mature either into B cells or they travel to the thymus and become T cells. Within the lymphoid system, of all the lymphocytes in the body, 10 to 20% are B cells, the largest majority are T cells, and about 22% are natural killer cells. And this slide is here. Just remember that T cells they grow up, they mature in the thymus, and surface antigens are acquired as lymphocytes travel from the bone marrow through the thymus, and they mature into T lymphs, and then they're reduced from the medulla of the thymus. T cells develop their identifying characteristics in the thymus, and then they're released from the medulla. Differentiation occurs once mature T and B cells are released from the bone marrow and the thymus. They migrate then to the secondary lymphoid organs and become part of a recirculating pool. So they recirculate all the time. And we've already talked about the secondary lymphoid organs. Lymphopoiesis, or reproduction of lymphocytes, occurs in the secondary tissue, and it is antigen dependent. And remember, from the bone marrow, we develop lymphocytes without antigen exposure, and once we get into the secondary tissue, our lymphocytes are developing based on antigens. And that makes sense. I like to think of it as lymphocytes are born, and then they have to go to school in the secondary tissue where they're exposed to new antigens, and they learn to be specialized. I like to think of it that way. So we call naive or resting lymphocytes that are just hanging out with nothing to do. Within a few days after they leave the primary lymphoid organs, unless they are activated by the presence of a foreign antigen, they die. And I like to think of this as they have nothing to do, and so they die of boredom. However, this process gives rise to long-lived memory cells and shorter-lived effector cells that are responsible for the generation of the immune response. The spleen is the largest secondary lymphoid organs. Its function is to, remember, destroy old red blood cells or filter out antigens from the blood. The structure of the spleen allows lymphocytes and macrophages to patrol constantly. So they are constantly patrolling or surveying for infectious agents or other foreign matter. Lymph nodes are located along the lymphatic ducts, and they serve as central collecting points for lymph fluid. This lymph fluid arises from passage of fluids and low molecular weight solutes out of blood vessel walls and into the interstitial spaces. So it's just like fluid from blood and tissues. The lymph nodes are especially numerous near the joints and where the arms and legs join the body. And if you remember back to the photo, the picture I just showed you, you could see those lymph nodes gathered in the joints and where the arms and legs join the body. The lymphoid system, the lymph node, is filtration. That's a main function of the lymph nodes. The lymph nodes contain sinuses, which are lined with macrophages, and that creates an ideal location for phagocytosis to take place. And you should note that when patients have a mastectomy, usually they remove lymph nodes too. So that's why when we are drawing blood, we need to avoid the arm that's closest to the mastectomy side because they don't have as many filters to catch bacteria. So that's why we need to be aware of that. So here is the picture of the lymph node. And just know overall that antigens go in through the efferent lymphatic vessel, and they go out through the efferent lymphatic vessel, and they can get circulated, and different B cells and T cells congregate in different areas of the cortex. but we don't really need to go into detail about the lymph node structure. The secondary follicles consist of the antigen-stimulated proliferating B cells. The germinal center is the interior of a secondary follicle where blast transformation of B cells takes place. Plasma cells actively secrete antibody. They're also called an antigen-activated lymph. That makes sense, right? They are making antibodies because they were activated by an antigen to do so. Memory cells are present in the germinal center of the lymph nodes, and this is where we get our B cell memory generated in our lymph node. Additional areas of lymphoid tissue include the MALT, that include the tonsils, the appendix, and the cult. We talked about that in the first lecture. And remember that Peyer's patches represent a specialized type of MALT, and they're located at the lower ileum of the intestinal tract. And these are bundles of lymph tissues organized in the ileum, so Peyer's patches. Make sure that you remember that term. The tonsils are another area of lymphoid tissue found in the mucous membrane lining of the oral and pharyngeal cavities. We know where those are. And an additional location of lymphoid tissue is the appendix. Within each of these secondary organs, T cells and B cells are segregated, kind of like police stations scattered throughout your body. B cells differentiate into memory cells, and plasma cells are responsible for humoral immunity and antibody formation. So the humoral immunity is the stuff that's circulating through your blood, or the humors. It goes back way into early medicine. T cells play a role in a cell-mediated immunity and produce sensitized lymphocytes that secrete cytokines. So, how can we tell B cells from T cells? Because I've only showed you one picture of what lymphocytes look like, and plasma cells. We can tell what the difference between B cells and T cells are based on markers on their cell surface. And these markers are called clusters of differentiation, or CD antigens. So, have you ever heard of CD4 and CD8? That's where that comes from, clusters of differentiation. And why do we need to know this? For example, we need to know what type of white blood cell or what type of lymphocyte is present. Just one example is in the treatment of HIV. If HIV virus attacks only CD4 T helper cells, then we need to be able to measure the presence or absence of CD4 cells versus CD8 or other types of white blood cells. Just know that there are a whole bunch of these CD antigens, and the only ones that we need to worry about are CD4 and CD8. Now, what do we want our B cells to do? We want them to make antibodies. So there are a lot of stages in B cell differentiation, but we are not going to get into the details of like pro B cells, pre B cells. We don't care about that. Just keep in mind that we want our B cells to make antibody. Do we want them to make just one kind of antibody? No. Therefore, we need B cell differentiation, right? Because we want to make all these different kinds of antibodies. And in our next lecture, we're going to talk about antibody structure and function. So this is going to make a little bit more sense. But I want you to pay attention to this image and notice that right here we have the pro B cell, and there's rearrangements of heavy chains. Heavy chains are part of our antibodies. And in the cytoplasm of this pre-B cell, you have mu chains. So mu are heavy chains. And then so the mu chains are the first ones that go to the outside of the cell. And we're going to learn in the next chapter about antibody structure and function that IgM with mu heavy chains are the ones that are made first. So there is a method to all this madness. Don't get overwhelmed. Just know that B cells go through a whole differentiation process, but what we need to know is that the mu chains are made first, and that makes total sense because IgM is made first in fighting diseases. Make antibodies. That's what we want them to do. Okay, so pre-B, the pre-B cell is where we start to see heavy chains made and the mu chains accumulate in the cytoplasm first. And at this point, pre-B cells are the only ones that express the mu heavy chains. The next stage is known as immature B cells, and these have complete IgM molecules on the cell surface. Isn't that fun? Variable regions also occur. We're going to learn about that next in the next chapter. And other proteins begin to appear, like the more CD antigens, but don't remember, all you need to know is CD4 and CD8. And also, this is where we start to get their MHC class II molecules. Oh, and this is going to be important for when we get to the complement chapter. CD21, you don't really need to know the number, but just know that these B cells have receptors for complement components. So file that away because there's a whole chapter just on complement. So immature B cells have CD40 and MHC2 molecules, which are important for interaction of B cells with T cells. That's the point. These B cells have to be able to interact with T cells. Remember the antigen presenting cells and the MHC class II molecules, it's all coming together. Many B cells, about 90%, are deleted from the bone marrow by the process of apoptosis. So they don't even get a chance to leave. So most of them just die. In the spleen, immature B cells develop into mature B cells. And then our mature B cells have IgD, which is another class of antibody. If a B cell is stimulated by an antigen, it will undergo transformation to a blast stage, and that's an early stage of cell development that we'll get to later on in hematology. This eventually forms a memory cell and an antibody-secreting plasma cell, and this process is known as the antigen-dependent phase of B cell development. Now, I do not expect you to memorize these stages of B cell development. Just know that they're the part in the blood, in the bone marrow. B cells, lymphocytes are going to develop whether or not there are antigens present. But once they get out into the secondary system, it becomes antigen dependent, because if they don't come into a contact with an antigen, they're just going to die. These B cells have a half-life of more than 6 weeks. We don't really care about this. All right, but we do care about that when B cells are activated, they give rise to plasma cells and so-called memory cells. And here is a lovely picture. This is a plasma cell right here. It's huge. It's busy making antibodies. And remember, what is a normal size for a lymphocyte? 7 to 10 or 7 to 12 microns. And plasma cells are about twice that size, 10 to 20, because they are busy making antibody. And antibody is also known as immunoglobulin. Plasma cells are not normally found in the blood, but inside the peripheral lymphoid organs. This is explaining memory cells that we don't really need to go into that. But memory cells are ready to make antibodies if they come into contact with an antigen that they've seen before. So now we're going to talk about T cells. T cell differentiation or maturation takes about 3 weeks. Remember, these are 60 to 80% of circulating lymphocytes in our peripheral blood, and the lymphocyte precursors are called thymocytes, and they enter the thymus from the bone marrow. This process is driven by chemokines, and we don't care what kind of CD antigens they have. But what we are interested to know is that maturation takes about 3 weeks as they're filtered through the different parts of the thymus. And it's estimated that 97% of the T cells die, but the survivors of T cell maturation have CD4 or CD8 markers where they migrate to the medulla of the thymus. CD4 positive cells are called T helper cells. They recognize antigen and MHC class II proteins. And CD8 positive cells are the T cytotoxic cells. They represent about 1/3 of the peripheral T cells, and they recognize antigen and MHC class I. And we already learned that already. Isn't that great? These mature cells are released from the thymus, and they seed these secondary lymphoid organs. Resting T cells have a lifespan of up to several years in these peripheral organs, and they can produce, certain T cells can produce things like interferon gamma or tumor necrosis factor, which protects cells against intracellular pathogens. Natural killer cells are the last type of lymphocyte that we're going to talk about. These are a very small percentage of lymphocytes, and they don't have the markers that T cells or B cells have. These are very large granular lymphocytes, and these are not the kind of granulocytes that we talked about with eosinophils, neutrophils, and basophils. Just know it gets hard to differentiate lymphocytes sometimes in peripheral blood. We're going to spend a lot of time working on that. But the important thing about natural killer cells is that they have the ability to mediate cytolytic reactions, so cell-killing reactions, without prior exposure. Natural killer cells lack specificity in their response, so this does not fall under adaptive immunity. Remember, this is innate immunity. And this gives time for the acquired response of specific T cells and B cells to be activated. So the natural killer cells, it's like, imagine the antigen comes in and the natural killer cell is covering for T cells and B cells. Natural killer cell activity is based on a balance of inhibitory and activatory signals, and the inhibitory signal is based on recognition of MHC class I proteins expressed on all healthy self cells. And we've heard that about 15 times already in the first couple of weeks of immunology. So if a natural killer sees MHC1 markers on a cell, it's marked as safe and it will not attack it. What is the mechanism of cytotoxicity? So this is asking, what do natural killer cells do with cells that don't belong? So diseased or cancerous cells tend to lose their ability to produce MHC proteins. So cancer cells don't generally make MHC1 molecules, so a natural killer cell can attack it because it is missing that self marker. And if an inhibitory signal is not received at the same time as the activation signal, then natural killer cells release substances called porphyrins and granzymes to destroy the cell. And this is an antibody-dependent cell cytotoxicity. Natural killer cells can also recognize and lyse antibody-coated cells through this process. So any cell coated with immunoglobulin or an antibody, especially IgG, it can be destroyed, and that is very interesting. And now I'm gonna post a really interesting video for us to watch.